Good morning. My name is Vanshika from BSc Chemistry Honors final year. In this audio lecture for visually impaired student, I have taken the topic proteins from book Pradeep's Organic Chemistry by Dr. S. N. Dhawan. So let's start with proteins. Proteins are extremely complex natural polymers and are rated first amongst the organic compounds essential for growth and maintenance of life. They are present in almost all living cells, being found in almost every part of every plant and animal. In human beings, they are the main constituents of muscles, skin, hair, nails, tendons, arteries, and connective tissues. In nutshell, each living cell is made up of thousands of different proteins. The protein present in different plants and animals, and even the proteins present in different tissues of a particular part of plant or animal, are different from one another in the composition and biological functions. However, all proteins contain the same element, that is carbon, hydrogen, oxygen, nitrogen, and sulfur. Some of these may also contain phosphorus, iodine, and traces of metals such as iron, copper, zinc, and manganese. All proteins on hydrolysis give alpha amino acids. About 26 alpha amino acids have been isolated from the hydrolysis of proteins, but the number of proteins obtained from these amino acids is very large because of the fact that there is no restriction on the number, types, and specific sequence in which the alpha amino acids could be present in a protein molecule. Plants alone can synthesize proteins from CO2, H2O, and inorganic nitrogenous compounds, but animals mainly depend upon plants for their protein requirement. Chemically, proteins are essentially polypeptides having molecular mass more than 10,000. Next is classification of proteins. Proteins have been classified in a number of different ways. The two most widely used systems of classification are discussed below. First, on the basis of molecular structure, proteins have been classified as first fibrous proteins and second is globular proteins. So fibrous proteins, these proteins consist of linear thread like molecules which tend to lie side by side to form fibers. The polypeptide chains in them are held together usually at many points by hydrogen bonds. As a result, intermolecular forces of attraction are very strong and hence fibrous proteins are insoluble in water. Further, these proteins are stable to moderate changes in temperature and pH. Fibrous proteins serve as the chief structural material of animal tissues. For example, keratin in skin, hair, nails, and wool, collagen in tendons, fibrin in silk, and myosin in muscles. Second, globular proteins. The polypeptide chain in these proteins is folded around itself in such a way so as to give the entire protein molecule an almost spheroidal shape. The folding takes place in such a manner that lipophilic, that is non-polar fat soluble parts are pushed inwards and hydrophilic, that is polar and water soluble parts are pushed outwards. As a result, water molecules interact strongly with the polar groups and hence globular proteins are water soluble. As compared to fibrous proteins, these are very sensitive to small changes in temperature and pH. This class of proteins includes all enzymes, many hormones such as insulin from pancreas, thyroglobulin from thyroid gland which produces the hormone thyroxine, antibodies responsible for allergies and defense against foreign organisms, hemoglobin which transport oxygen from the blood to the muscles, fibrinogen which is converted into the insoluble protein fibrin and thus causes clotting of the blood, albumin in eggs, etc. Each globular protein has its own characteristic geometry, which results through the interaction between different sites in the same polypeptide chain. These interactions are usually of four types. First, disulfide bridging. Second, intramolecular hydrogen bonding. Third, van der Waals interaction. Fourth, dipolar interactions. So, globular proteins may consist of a single polypeptide chain or may consist of several polypeptide chains called subunits or protomers. Each subunit has its own three-dimensional geometry. The various subunits are then held together by secondary forces such as hydrogen bonding, van der Waals interaction, etc. to give the molecule its overall spheroidal shape. Second, on the basis of chemical composition or the nature of products they give upon hydrolysis, proteins can be classified as follows. First, simple proteins. Second, conjugated proteins. So simple proteins on hydrolysis give only alpha amino acids. For example, albumin in the white of eggs, glutenin in wheat, oxygenin in rice, keratin in hair, nails, horns, etc. Conjugated proteins on hydrolysis gives a non-protein portion in addition to the alpha amino acids. This non-protein portion is called the prosthetic group. The main function of the prosthetic group is to control the biological function of the protein. Depending upon the nature of the prosthetic group, the conjugated proteins are further divided into a number of different types. Like the protein nucleoproteins, its prosthetic group is nucleic acids. Glycoproteins or mucoproteins, its prosthetic group is sugars. Lipoproteins, prosthetic group is lipids such as lecithin. Phosphoproteins, prosthetic group is phosphoric acid residues. Chromoproteins, that is hemoglobin and myoglobin. Some coloring matter such as red colored protoporphyrin. Third is derived proteins. These are the degradation products obtained by partial hydrolysis of simple or conjugated proteins with acids, alkalis, or enzymes. For example, proteoses, peptones, and polypeptides. General characteristics of proteins. First, elemental composition. The elements generally present in proteins are carbon, hydrogen, oxygen, and nitrogen. Their approximate composition is almost always carbon 53%, hydrogen 7%, oxygen 23%, and nitrogen 16%. Sulfur and phosphorus, when present, occur to the extent of 1% and 0.5% respectively. Second, high molecular mass. Proteins have very high molecular masses ranging from 10,000 to over 50 millions. The reported molecular masses of some proteins are egg albumin 34,000 to 44,000, milk albumin 35,000 to 40,000, hemoglobin 65,000 to 70,000, and casein 75,000 to 1 lakh. Physical state. Proteins are generally colorless amorphous solids having no definite melting point. However, some of them have been obtained in the crystalline state. They are usually colloidal in nature. This property of proteins to readily form colloidal solutions with water has been used to separate them from soluble crystalline salts by the process of dialysis. Fourth, amphoteric nature. Like alpha amino acids, proteins also exist as zwitterions or dipolar ions and hence react both with acids and bases. Fifth, isoelectric point. Like alpha amino acids, each protein has a characteristic isoelectric point, that is pH at which its ionization is minimum and is least soluble. For example, casein has an isoelectric point of 4.6 This property of proteins is often used for their isolation. Sixth, optical activity. All proteins are optically active, and further, all of them have L configuration since they are made up of L amino acids. Hydrolysis. All proteins can be hydrolyzed by acids, bases, or enzymes. The hydrolysis proceeds slowly and in stages, ultimately giving alpha amino acids. The sequence of hydrolytic products is shown below, in which each hydrolytic product is less complex than the preceding one. Proteins, it is converted to proteoses, then peptones, then polypeptides, then simple peptides, and finally into alpha amino acids. Conjugated proteins also yield products other than peptides and alpha amino acids. Next is biological functions of proteins. Proteins constitute an indispensable part of our food. These are required for building new cells and repair of worn out ones. Obviously, children and expectant mothers require more proteins, but before they can be utilized by the body, they must be hydrolyzed to the constituent amino acids. Hydrolysis of proteins begins in the stomach, where HCl causes denaturation of proteins. The denatured proteins are then hydrolyzed by the enzyme pepsin into lower molecular mass polypeptides. In the intestine, the enzymes trypsin and chymotrypsin complete the hydrolysis to alpha amino acids. The alpha amino acids are then assimilated in the bloodstream and are carried to the cells in the various parts of the body. There are some of the amino acids are converted back into specific proteins which are required by the body for its growth and maintenance, while others get burnt as fuels to supply energy. Thus, proteins are being constantly broken and resynthesized in the body, and this continuous dynamic process helps to fulfill the needs of the body. Having discussed above as to what happens to the proteins in the body, let us now discuss some of the important biological functions of proteins. Some of the important biological functions of proteins are listed below. First, structural materials. One of the most important function of protein is that they serve as the chief structural material of animal tissues. Structural proteins are approximately one half of the body's total protein content. Some common examples of structural proteins are keratin in skin, hair, nails, wool, horn, feathers, etc. Collagen in tendons, myosin in muscles, fibrin in silk, etc. Second, transport agents. Certain proteins serve as transport agents or delivery vans in the body. They transport the cellular necessities such as oxygen, metals, fatty acids, hormones, etc. For example, hemoglobin present in blood transports oxygen from lungs to all the parts of the body. Third, enzymes. These are globular proteins which act as biological catalysts. Their action is very specific and highly efficient. They catalyze the reactions in aqueous solutions without any change in temperature or pH. They can bring about breakdown and synthetic reactions much more rapidly in the living organisms at body temperature than is possible in the laboratory, even under drastic conditions. For example, the enzyme carbonic anhydrase present in the RBCs can catalyze the decomposition of over 36 million molecules of carbonic acid, that is H2CO3, to CO2 and H2O in 1 minute. The reverse reaction, that is the conversion of CO2 and H2O into carbonic acid, is catalyzed by the enzyme carbonic hydrase. This reaction helps in maintaining CO2 level in the body fluids and tissues. Similarly, enzymes pepsin, trypsin, chymotrypsin catalyze the hydrolysis of proteins into amino acids in the digestive tract. The enzyme ptyalin present in saliva catalyzes the hydrolysis of starch into maltose during the chewing of food in the mouth. The enzymes maltase, lactase, sucrase, and amylase catalyze the hydrolysis of di and polysaccharides into monosaccharides. The deficiency of even one enzyme in the body can either slow down or prevent a particular reaction in the body and thus cause some serious disease. For example, the congenital disease phenylketonuria is caused by the deficiency of the enzyme called phenylalanine hydroxylase. Similarly, the disease albinism is caused by the enzyme tyrosinase. Fourth is metabolic regulators. Some globular proteins Let's again start with metabolic regulators. Some globular proteins regulate metabolic processes. For example, the protein hormone insulin from pancreas maintains glucose level in the blood 60 to 100 mg. The glycoprotein thyroglobulin from thyroid gland helps in the synthesis of hormone thyroxine. Fibrinogen, which is converted into the insoluble protein fibrin and thus causes clotting of blood. Nucleoproteins, constituents of genes, carry genetic information from parents to the offsprings. Fifth is antibodies. These are proteins which defend the body against the invasion of foreign organisms. When an infectious species, virus, bacteria, or a foreign protein enters the blood, it releases foreign substances called antigens. In response to these antigens, the body proteins produce specific proteins called antidisease. The antibodies thus produced then specifically combine with the antigens which induced their synthesis, thereby protecting us from disease. Gamma globulins present in blood plasma are example of antibodies. From the above discussion, it is apparent that proteins perform a variety of biological functions. Therefore, they constitute an indispensable part of our food.